Hey guys, trust everyone is doing well. I'm Nimra Mehmood, recent graduate from Dow University of Health Sciences. My career in research begins from Novartis Pharmaceutical. Then I'm currently working as Central Clinical Data Coordinator at Matrics Research Private Limited, as well as I'm enrolled in a Certified Clinical Research Program. So I just want to keep you guys, before just switching towards my presentations, because as per my understanding, I am assuming that most of the audience here is actually graduated or undergraduated students. So before directly jumping into my presentations, I want to keep you guys apprised regarding basic insights, like crux of the meaning for my entire graduation journey. Because at this point in time, I feel like students are like real demotivated. So I do want to share a crux of the meaning for my entire journey, and then I'll switch towards my presentation. So, but I feel like in final year, pupils literally crave for outcome. They don't want to be part of that process to achieve that outcome. But it's that process which literally makes you feel, which literally makes you realize whoever is worth sharing your table with. And as just to make sure you guys are on the same page, I feel like in final year, I literally think that I'm surrounded by negative energy. And like, nobody literally want to help me out. It's like 80% of people don't want to help you out, but still there are 20% of people who want to help you out. And it's very great insight that you still have those 20% of people in your life. If I give my example, the person sitting here, Sir Arif, I'm having my lecturers as my mentors. In a professional resource setup, I'm having my project manager, my team lead. These people are literally working tremendously hard on me, despite the fact they're having no personal benefits, but still. So finding these people in your life is literally hard, grateful. And once you find these people in your life, just ask them their experience, literally are here to guide you. So that was the crux of the meaning for my entire journey. I guess all of you are literally having those 20% of positive pupils around you who literally motivate you despite of any other thing. So I guess now, just noticing in time, I just want to proceed towards my presentation. The topic of my presentation is Understanding Clinical Research Trials for Allied Health Perfusion Students. Okay. Moving forward. Okay, perfect. Thank you. Okay. As being medicos here, we are apprised regarding basic terminologies, like what is medical terminology. We are apprised regarding what is research. The final 2 years of medical technology are actually research-oriented. So we guys are apprised regarding basic research methods and terminologies, study design, study techniques, for sampling techniques. For example, we are having simple random sampling, we are having a stratified, cluster, snowball. We are apprised regarding these things. This is actually a ground foundation which can provide you all with the required boost of thrill you need to understand clinical trials. So without wasting any further time, because we guys are on the same page regarding research. So I'm just quickly switching towards the specific things you guys need to know for clinical trials. So there are certain, I'm currently switching towards guidelines and practices. So there are certain basic internationally approved guidelines and practices that you guys need to have a precision of. These guidance, strict adherence to these guidelines is really important when you are working in any professional clinical trial setup. The first guideline is ICH-GCP. This is International Council of Harmonization for Good Clinical Practice. This is actually ethical scientific guideline to conduct any trial that involves human as subject in order to protect the rights of humanity. Then second, we are having GDP, Good Document Practice. This is also an international scientific guideline to have rigorous control over documentation, because everything in clinical trial is being documented. So we need to have rigorous control over documentation. For example, if we need to correct any data points, you can't literally cross that thing. We need to make a straight line and we need to sign your initials over that. Signing proper documentation, make appropriate corrections to accurate data points is really important, because everything in clinical trial is being documented. So you need to have rigorous control over your documentation. The third thing is HIPAA. It's also actually an international scientific guideline for protection and privacy of medical records for patients. Because there are literally some sensitive patients, the medical records should be protected. So there should be like privacy for that patients. Then we are having ALCOA. Well, I'm specifically talking about ALCOA. I am literally referring to all these guidelines, like GCP, GDP, every guideline. Because documentation should be attributable, legible, contemporaneous, original, complete, and accurate. So all these guidelines and practices are being implemented when you are working in a professional research setup. So strict adherence is really mandatory if you are working in a clinical trial setup specifically. Okay, after these guidelines and principles, now we are delving into the types of clinical research trials. So when I'm specifically referring to clinical research trials, I'm just talking about two things here, drug or any new device. The clinical research trials are basically based on this drug or device. So clinical research trials are actually scientific research studies that involves human as subject. And what are we finding in these clinical trials? We are finding the dose or drug or device safety, efficacy, and effectiveness. Before these drugs are being released into the market for public use, we need to check the safety, efficacy, and effectiveness of that drug, and for that purpose, we are conducting that clinical trial. Now, I'm spilling some tea here. Every clinical trial is having a protocol and an ECRF guideline, which is specifically for that clinical trial. What I mean by protocol, everything in that clinical trial is being recorded in a separate document, which is known as protocol. Everything related to that trial. For example, beginning with synopsis, to research methodology, study design, study technique, sampling technique, adverse events, concomitant medications, medical history, primary and secondary endpoints, what interventional drug or device is being provided to that patient. Everything is being documented in that protocol. And strict adherence is really necessary for that protocol, otherwise you need to record a protocol deviation. That literally doesn't sound good to me. Then we are having ECRF guidelines. In ECRF guidelines, everything regarding data is being mentioned. For example, data collection, data entry, for each and every subject, we need to enter data in electronic portals, that is EDC portals. So you need to, in ECRF guidelines, everything related to data is present. For example, audit trail, how to enter the data in EDC portals, and regarding data cleaning, data validation, quality control of the patient's data, everything is mentioned in ECRF guidelines. So each and every clinical research trial has its own protocol and ECRF guidelines, and strict adherence is really necessary. Otherwise, you need to record a protocol or ECRF deviation. Okay. So as per this time, we know about like what are basic principles, what are basic guidelines, what is clinical research trials, what are protocols and ECRFs. Now we are moving toward the phases of clinical trials. So first we are having preclinical phase, which is actually animal trials, and then we are moving towards clinical phases. In clinical phase, we are having 5 phases, phase 0 to phase 4. In each and every phase, there are certain primary and secondary endpoints you really need to focus. So for phase 0, which is actually microdosing phase, the only concern is safety and safety. Pharmacokinetics and pharmacodynamics, that's it. In phase 1 clinical trials, what we usually notice, like the primary concern is safety of the patient, because now you're interventioning that drug into that patient, so the primary concern is just safety. Then phase 2, the primary concern is safety, but along with safety, you're measuring the dose titration, the dose efficacy, basically the drug efficacy and dose titration is the primary endpoint for like phase 2 clinical trials. Then proceeding towards phase 3 clinical trials, now we have done with safety, dose titration, so we need to focus on drug efficacy and effectiveness. Then finally, we are having phase 4 clinical trials. So in phase 4 clinical trials, we are focusing on post-marketing surveillance, because at phase 3, after the approval of phase 3, that drug or device is being released into the market. So at phase 4, we are watching the post-market surveillance, that's it. So in all phases, there are certain primary and secondary endpoints which we are noticing. And these endpoints really matter. Okay, here comes my favorite part, like visitor flow or the flow of subject in clinical trials. Like how actually a subject enters into the clinical trial, how actually a subject completed a clinical trial. For example, we all are cardiac pupils here, like related to cardiovascular perfusion. So let's just take an example of any cardiac trial. Consider there is a trial, clinical trial related to cardiovascular diseases going on. And there is actually a physician who is principal investigator for that trial. Okay, let's just make it very simple. Assume a patient comes in OPD, and that patient is having a consultation with this physician, and that physician is actually a cardiologist actually. What happens here is that during that patient's consultation, because that physician is actually a consultant for that patient, but also that physician is having a clinical research trial on that cardiovascular disease. So during the patient consultation, the physician is actually pre-screening that patient for the clinical trial. For example, there is any clinical trial going on cardiovascular disease, and that patient is having the primary disease endpoint, which is cardiovascular disease. So during that consultation, he's actually using the protocol things, that's inclusion and exclusion criteria, and he's actually pre-screening that patient if that patient is like suitable candidate for that clinical research trial. Then after this consultation, that physician goes and asks to the patient that I'm actually a principal investigator of clinical trial, which is actually being focused on that primary condition you are having. So are you interested to enroll in that clinical trial? If that patient is really interested to get enrolled in clinical trials, he gives that physician a green check. Yeah, I'm good to go. What happens later is that physician, who is actually a principal investigator for that trial, that physician connects that subject with clinical research coordinators. These are CRCs. What clinical research coordinators do, they have excellent adherence of protocols, of ECRF guidelines, so they guide that subject, that patient, regarding everything in clinical research trial. They give ample time to subject to sign informed consent. Once informed consent is being signed, now the procedures start. Now, here the interesting fact is that in protocols, we are having SOA, schedule of activity. Consider we are having any trial on cardiovascular disease, and in that cardiovascular disease, as per protocol, we are having 10 visits for that patient. Under each visit, there is some monitoring, specific monitorings as per protocol, which are mandatory to perform. I'm assuming if that patient signed informed consent, he's ready to get enrolled in that trial. So in visit 1, in a screening visit, as per protocol, the things, for example, inclusion criteria, exclusion criteria, eligibility, if that trial is cardiac specific, then I'm literally assuming there would be any cardiac specific things. For example, ECG, we could have cardiac biomarkers, some central labs, some local labs. These assessments need to be performed, like a prohibited medication, con meds, and after that first visit, that principal investigator and monitor of that trial will decide if the subject is eligible to get enrolled in the study or not. If the subject is fitting the criteria for that study, he's having a green check. Now he can move for different visits like visit 2, visit 3 to end, like visit 10, end of treatment visit. And what happens is, if the subject fills inclusion criteria, he's being scheduled for second visit, then onwards all the visits are being completed. But what if the subject didn't fit in the clinical trial, for example, doesn't qualify the first screening visit? That subject is considered a screen fail and he's not allowed to proceed further. But if that subject qualified the first visit, that screening visit, he's being scheduled for the second visit, which is actually baseline visit. And in that baseline visit, the subject is being randomized. He's giving IP investigational product for which the research is being going on. And after that, the subject is having their own choice if they want to complete that clinical research trial or whether they can withdraw consent at any point in time. Because honestly, the rights of humanity are being protected when we're specifically talking about clinical research trial. So that subject can withdraw consent at any point in time and can be marked as discontinued subject and in a clinical research trial. everything at every step is being documented in EDC portals using ECRF and protocol guidelines. So there's a strict adherence to ECRF protocol guidelines. There's a strict adherence to ICH, GCP, GDP, every compliances in every phase of clinical research trial. So I guess that's it. I kept you guys apprised regarding basics of clinical research trial. If there would be any current hesitations, you guys could reach me out anytime. Perfect. Thank you.